Hello, Professor Stephen Harris. You are a botanist and associate professor in plant sciences at Oxford University at Christchurch College, is that right?
Yes. Yeah.
So thank you very much for sparing the time to have a chat to me today and explain some of the things that I like to think about in terms of flowers and drying flowers and the processes and things like that. So if you don't mind starting by just introducing yourself and saying who you are and what you do.
Yeah. So, well, as I say, I'm Stephen Harris, and within the university, I am the curator of the Oxford University Herbaria. So I'm responsible for a collection of herbarium specimens, squash dried plants, approximately 1 million specimens that date back to the late 16th century.
Brilliant. So, and where's that? Is that used within the university?
It's used, yes, it's used very extensively within the university. So we're located within the Department of Plant Sciences on South Parks Road, but the collection is used both by our own researchers within the department, by researchers and students across the university for both research and teaching, and it's also used internationally as well. So we've got a lot of international collaborators from a whole range of different disciplines, not just botanists, but across academic disciplines that range from sort of archaeology and English and philosophy, history, through to geology, engineering, zoology. So it's very extensive.
Yeah. Well, gosh, the biggest surprise there was engineering, I suppose, but I suppose that does make sense.
Yes, so people are interested in plant structures and how how plants are put together, the sorts of materials that plants are made of, and how all those elements come together in order to produce essentially plants that can that are adapted to a whole range of conditions that they face within their natural environment.
Okay, so I'm just going to fire a few questions at me. As I said to you before, I'm a bit of a lapsed biologist turned florist. So I can't help but think sometimes about the sort of biology behind the flowers that I use, and particularly what happens to them when they're dried, because I specialize in dried flowers. I work with dried flowers, I press flowers, and it always interests me sort of what's actually going on and why some flowers and plants dry so well and some I at least can't get to dry very well. So perhaps first of all, would you mind explaining what actually happens when flowers and plants are dried or pressed? Like what, apart from, I mean, obviously they lose their water, but is there anything else that happens?
Right. So there's a number of things that are going on. So I think the first thing to think about in terms of a plant is that you can think about it as being constructed of a whole series of boxes, and those boxes have a wall around them, and that wall is made of a whole series of substances, particularly cellulose. And within that, we've got essentially a bag full of water, which is the cytoplasm and the nucleus and everything that makes the sort of the cell work, if you like. So all of these are stacked together in all sorts of different arrangements, so that when you dry something, essentially what happens is that you lose the water. So the bag in the middle actually contracts and it gets smaller. And if the wall of the box isn't tough enough and to support that cell, once you've removed the water, it collapses. So the whole thing shrinks. And so this is what's happening fundamentally when you dry something. You're removing that water, and you've got the issue of therefore the cell, if you like, collapsing. So what you're trying to do then when you dry a plant is to provide that support in order to prevent the cell collapsing until the cell is dried. So imagine a piece of paper. A piece of paper, when it's dry, is really stable. When that piece of paper is wet, it breaks apart very easily. So you can imagine a plant as being like that piece of paper. That cell wall around that box is like that piece of paper. So in the early stages of the drying process, it's supported by the water within it, within the cell. That as the drying process continues, that cell gets wet, the wall gets wet, but there's no support on the inside. So if you just dry it without any external support, usually the whole thing just collapses. It just shrinks. So when you put something under pressure in a plant press, what you're trying to do is you're trying to provide that support until the plant is dry enough to be able to be like a piece of dried paper. So that's what's going on. So, but of course, the other way of drying plants is not under pressure. So there's a whole group of things that will survive very well without that pressure. So I see that there's a whole series of dried plants behind you,
yeah.
And they've all been dried without pressure.
Mm.
But in this case, what you've got structural support provided by the plant itself. So there are things within the cell walls of the plants. There are chemicals that thicken the cell walls, like lignins, these types of things. I see you've got some grasses there. Grasses will have cell walls that have got lots of silica in them, lots of sand in them, which provides really good support. And then the other things that are supporting plants will be some of the internal structures. So some leaves, for example, have lots of they've got lots of vascular tissue in them. That vascular tissue has got lots of support structures around it. So in that case, it's a bit like reinforced concrete. And you've got these support structures actually running through the structure itself, so that when you dry the plant, although the individual cells collapse, everything, the overall structure remains. So those are the fundamental things that are going on.
So I suppose just to go back and clarify what you just said, which is really interesting, which is, so the things that dry well and fairly easily, some of them dry without any effort at all. They almost seem to dry, while they're, just as part of their life cycle. Things like poppy seed heads and grasses.
Yes.
They have their cell walls or internal structures have particular chemicals or properties. That grasses have silica in them, which is like sand.
Yeah.
And so that stops the cell walls collapsing when the bag of water inside shrivels and disappears.
Yeah, or it provides sufficient support for the cell wall while that cell wall is drying.
Yeah.
So again, it's like the scaffolding that you might have around when you're putting a building up. You want that scaffolding there to actually support the structure. But if you like, when the whole thing is dried, then the cell walls are hard again, if you like, but they've had this additional scaffolding support around them.
So is this why it's often easier or better to dry flowers upside down, so to turn them and hang them upside down, so that they don't have I suppose in general, the heaviest parts of the plants are at the top, the flower heads or the seed heads. So if you turn them upside down, then they're not sort of succumbing to that pressure of the heavy thing on top while that's all caving in.
Absolutely. So you can, if you imagine going back to your poppy example, if you were to dry a poppy sort of upright, then you've got the junction at the top of the poppy head and the stalk, and as that dries, that will get very floppy, and then the whole thing will just fall down. Whereas if it's upside down, then you can, that junction will still get floppy, but you've got essentially gravity there keeping it straight until it's sufficiently dry that it becomes nice and stiff, and then you have your upright poppy heads. And it's the same for all of these other examples.
And is there an evolutionary benefit to drying well, to a plant being able to dry out like that?
So so obviously it depends on there are different sorts of habitats where you might expect to find plants which dry, if you like, well from our point of view. So for example, if you go into the Mediterranean, then there's whole forests of these of plants which are different sorts of forest plants with these very hard leaves. They're called sclerophyllous leaves, and you also find them in places like Australia, for example. So, for example, eucalyptus is an example of sclerophyllous leaves. So these are these tough, rigid leaves, and they're in environments where naturally the plant would potentially lose quite a lot of water and is exposed to very hot conditions. And what you don't really want is every time the sun comes out, your leaves collapsing. So you've got that support that provides that rigidity. So that's one condition where you might expect to find plants, if you like, that dry well. The grasses are another example of that, because most grasses are dispersed either by wind or by animals, and the flower heads are dispersed when they're the seed heads are dispersed when they're dried. So it's great essentially to be able to have those flower heads actually upright, so that either the wind can catch them or the animals can catch them as they're moving by. And so that works very well from that point of view. Of course, one of the disadvantages of a lot of grasses is that naturally the fruits break up. So from the flower head, because of course that's all about dispersal. So there's a whole group of grasses that would potentially look very pretty from a dry plant point of view, but the problem is that the flower heads themselves, they break up naturally. They dry very well, but they break up. All the bits drop off, which you obviously don't really want.
Not for my no.
And then other situations that you might have cases where you've got plants that are drying very well. Again, conditions where things are water stressed, where you've got perhaps short growing seasons. So examples of that might be helichrysums. So the helichrysums grow they dry without you actually doing anything at all, so in those conditions, a lot of the helichrysums are high elevation species. So they occur in areas where you've got perhaps water stress, you've got short days, you might have to expose to high temperatures. All of these types of things might be associated with features that provide you with conditions that allow you to sort of dry these plants effectively. And I suppose at the other extreme of that, there are plants that are really bad to dry. So for example,
because why won't they dry?
So a classic really bad plant to try and dry would be something like let's say a cactus. So cactuses are essentially they're just they're made up of lots of little bags of water. So they're made up of lots and lots of cells that have got lots and lots of water in them, but they've got very little. There's very little membrane around it. The walls are very thin in a cactus. So when the whole thing is when the whole thing dries, then you lose this massive amount of water, and they really do, they really shrivel up into something absolutely tiny, and also they dry very slowly, because one of the things about cactuses is it's not just, if you like, the water that's in them, but also, in order for them to be able to survive, they have the cells contain lots and lots of polysaccharides. So there are lots and lots of mucilages and this type of thing. And plants have got lots of mucilage in them, dry very slowly, and essentially it's a way naturally for the plant to hang on to water. So it's rather like these beads that you can put into houseplants, the soil of houseplants, to conserve water, and so that you don't have to water them quite as often. So it's this type of thing. So they're gels that essentially hold on to water. So plants like this are really bad to dry.
And you'd almost think that because cactuses and succulents and things have such, we're sort of taught at school, I suppose, about them having really thick, waxy skins to keep all the moisture in. But you're saying that it's the cells inside that have really thin skins.
Yeah. Yes. So certainly cactuses do have very thick cuticles on the outside of them. These layers, and those layers do provide a lot of support. So you can dry the outside of a cactus really well, and everything. It's just the internal bit that's the problem.
You just stuff it with something to.
Yes. Yeah. Well. Absolutely. I mean, if you're going to dry cactuses for things like herbarium specimens and this type of thing, if you take your normal barrel cactus, what you do is you chop off the top, chop off the bottom, you put a slit around down the outside, and then you would unroll the cactus. So it's just the skin, scoop out the inside, and then dry it like that. So you have three different bits. You have the outside bit, you have the top, and you have the bottom. Because the bit in the middle is you're never really going to dry it very effectively.
Yeah, well, that sounded like the beginning of a good recipe. So, one of the things that, or one of the flowers or plants that fascinates me is hydrangeas, because as a cut flower, I know as a florist, they're incredibly thirsty. They can't be out of water. When florists buy them in, they have a little sort of sock of water around the bottom of the stems, because unlike the other flowers that arrive, they can't be out of water for any time at all. And yet they dry so well. And to me, that seems a bit paradoxical.
Right. So of course, as we were just talking about, they've things like hydrangeas, they do have cuticles on them, and so they've got these waxy deposits on them. They've also got a lot of this sort of internal scaffolding that helps maintain their structure and maintain their form. They probably they're probably losing large amounts of water because most hydrangeas come from habitats where water is not really a limitation. So they're probably well adapted to actually just having water almost flooding through them, and it might actually be that some of them even have issues with having too much water. So you have to just be able to get rid of the water. But certainly in terms of hydrangeas, then in terms of drying, it's probably to do with the internal structures and things like cuticles and this sort of thing.
So one of the things that I think is an interest of yours, and also I find really interesting, is the the history of well, the history of botany is something that you are interested in, and I am interested in the way that pressings of flowers were used, you know, back in the, I suppose, the beginnings of botany and the sort of exploration in order to bring them back safely to study them. And your background, you were telling me just before we started. Can you explain what that is?
Yes. So, so the background that I've got is actually it's a sheet, a specimen of a fungus that was collected in Germany in the 1840s, and it was prepared by taking the fungus and cutting it into very thin slices. So you think about fungi, and you think about the fact that they've got massive amounts of water in them, and then when they dry, you know, most fungi, when they dry, they sort of shrivel into something, you know, into tiny little things. You know, you just have to think of dried mushrooms in your kitchen cupboard and this type of thing. They don't look very much like the mushroom that was actually growing in the field. So in this case, what they've done is they've taken very, very thin slices, just a couple of millimeters wide vertically, using essentially a cutthroat razor. And then they put these slices into a plant press, and the plant press is providing the support so that when the plant is dried, then it is actually it's then like a sheet of paper, and it's flat. And in this case, it was pressed onto the sheet of paper in this position, so that if you like, it's stuck onto the paper with its natural glues, and so that's what's going on here.
So are plant samples still preserved and used scientifically in that way, or are you still just using all of those you know, the historically collected samples? What happens now when botanists are collecting flowers and want to preserve them? Do they still use sort of pressing techniques and drying techniques, or is it all chemical?
No, it's pressing techniques. The techniques that are being used are the same techniques that were being used 450 years ago. You know, the kit may have changed slightly. You know, it's no longer sort of piles of rocks on piles of paper. You know, you have plant presses and this type of thing, and you know, there are techniques for drying these things quickly, but actually collecting dried specimens is still the fundamental thing that a lot of field-based botanists do. So we're adding specimens to the collections all the time, and there are hundreds of millions of specimens in the world's herbaria, and these numbers are growing all the time, because this is the fundamental research material for basic biodiversity research. So almost everything we know about the distribution of plants, the identification and the structure of plants comes from specimens that are contained within herbarium. And they act as this sort of long-term sort of repository of information. So one of the things about herbarium collections, particularly old collections like the one I'm responsible for in Oxford, we can go back 400 years to specimens, and we can see how things have changed. And of course, it's not just, we don't just have a specimen of each species. We have specimens, we hope, that are going to represent the distribution of that species and how that species varies in time and in space. And of course, you know, there's 2,500 herbaria worldwide, and we're all in contact with each other. So we're this great global database. And of course, these collections are now being digitized as well, so that they're even easier for people to get access to.
Yeah. And so the plants that you have, which are hundreds of years old, they're still useful for, is it still for looking at the structures, or are you looking at the genetics of them? Can you use them for genetic work as well?
We can, absolutely. So they're used in all sorts of different ways. So from a genetic point of view, you can extract DNA from these specimens. Depends on how they're dried, depends on the species and everything. But you can extract DNA from species that are hundreds of years old. You can extract secondary chemicals. So you can extract different sorts of chemicals that things like tannins and this type of thing. You can look at things like different isotopes, and you can work out perhaps where these plants were distributed, and therefore what the chemistry of the soil was that they were growing in. All of this type of thing gives you perhaps information also about what the atmospheric levels of atmospheric pollution may have been, for example, in plants. If you're dealing with aquatic plants, then it can tell you something about the water quality that these things were growing in. You can with herbarium specimens, you can sort of reconstitute bits of them, but for example, you can boil up flowers, and you can then look at the detailed structures of flowers. You can extract pollen grains from the anthers, and use those pollen grains perhaps to look at to be able to use them for identification purposes for what's going on in pollen cores, taken from lake beds to see how vegetation changes. You can look at how the morphology of a plant might change. You can look at how distributions of plants might change, for example, perhaps in response to climate change or in response to human activity. You can look at changes in population structures. You can see what sort of pollinators were being involved, how seeds were being dispersed. So there's a huge range of different things that you can do with these preserved collections.
Yeah, so they're real sort of time capsules. And I suppose in lots of ways, plants are the kind of, what's the word, like they give so much information about what's going on in general, that, you know, in, like you say, all of those different things. They can be kind of canaries in the coal mine for certain things, and give an idea of not just what's happening with them, but with everything, because they're interacting all the time with the different habitats and the ecosystem and the atmosphere and things.
Absolutely. So they can be really good environmental monitors, plants, and how plants are changing. And of course, these collections can also be used in terms of how we interact with plants and how plants interact with us, because it's a two-way thing. You know, plants change in response to us, but we change in response to plants as well. So we can look at these sorts of things. So we can, you can see trends, but in terms of what plants are becoming popular. Where do they become popular? When do they start to appear as part of human cultures? So when do we start, for example, cultivating certain things within our gardens and all of this type of thing?
And how long is there a limit to how long a plant sample, a pressed or dried plant, can last and be viable and useful, or does it depend?
Well, from the experience in our collections, and we have material that goes back to 1595, which is still useful. But of course, we know from archaeological deposits and archaeological sources that plants can survive a a very long time in their dried form, as long as you can keep insects and fungi away from them. So for example, the most famous of these are probably the dried flower wreaths that came out of the excavation of Tutankhamun. But you find them also in other conditions as well. You find them in at Inca sites all over the place. So these things can last for thousands of years, and we just need to look at a wood sample. A wood block is just it's another sort of dried plant. And those can last for thousands and thousands of years. And of course, with wood samples, of course, if they're underwater and everything in an anoxic environment, then they can be very well preserved.
So anoxic without oxygen, so without not exposed to the air. Yeah. Gosh, so it's really, I guess we'll find out in another few hundred years how your herbarium species are doing and whether they're still doing.
Well, yes, one hopes that that they will be here in 400 years' time. I mean, this is the 400th anniversary of the Botanic Garden in the university, and one would hope that in 400 years' time these collections are still here, and that they're still being used. And I imagine they will be being used in manners that we can't even conceive of, let alone anything else. So, because you can imagine that sort of 50 years ago, the ways that we're using collections now would, people would just not have even thought it was possible.
Yeah.
So they represent, they represent capital that we can use in all sorts of different ways, and it's not only that they were collected in the past, we're using them in the present, but they've still got a huge potential in the future.
Yeah, isn't that wonderful to think about the people who collected, who were going off on months and months of sort of exploration voyages, collecting these pressings on sailboats and bringing them back, and they potentially will still be used in hundreds of years to come, but for things like this, I mean, the things that we use them for today, the kind of engineering projects that people might be looking at them for, they wouldn't have even imagined those things. So how wonderful that their work still kind of lives on and feeds into all the technological advances that we enjoy. That's such a nice thing. It kind of makes me want to press more flowers and keep hold of them just in case they're useful.
Yeah, I mean the important thing, and I completely agree with that. Having the opportunity to handle and use and with this type of material, it's quite humbling to just think how the difficulties that individuals faced in order to move that specimen to the cupboards in Oxford over hundreds of years. And of course, the important thing with the specimens that are contained within the collections here is it's not only, if you like, the specimen itself, the dried plant, but it's also the information that comes with it. So if you like, it's that metadata. It's information about who collected it, where it was collected, when it was collected, how it was collected. All of this is really important in terms of being able to use that specimen. So, specimens have greater value from a scientific point of view, the more information that they have that accompanies them. Mm-hmm. And also, in fact, the more people that have used them. So, it's not just the fact that there's a collector may have collected this specimen, but it's generations of botanists have actually used this same specimen and have used this in terms of putting together their ideas and thinking about and the way that they consider plants.
And are all of their findings sort of linked back to that sample, so that if you're working on a sample, you can see what everyone else has sort of found out from it beforehand?
Yes, yes. So herbarium specimens often will have lots of labels on them. You know, they'll have labels that span maybe 100, 200, 300 years of different people's views. So they can become sort of, if you like, archival documents in themselves. And sometimes you can actually see academic debates being argued on these sheets as people agree and disagree on what's going on, and politely or less politely, depending on the individuals involved. So they become quite interesting social documents as well.
Yeah, how interesting. Gosh, I never would have thought. I mean, I knew that pressings used to be taken for botanical research, but sort of nowadays I just think of flower pressing as a hobby and something nice and something.
No. That's actually a problem for collections from a research point of view, is that when you talk about this to people in a general sense, the first thing they think of are Victorian ladies of a certain background doing this sort of thing in their in their spare time, or it's something that children do, and that's it. But of course, if you like, the techniques that the Victorian ladies and the children use are the same as we use, but if you like, the purpose and the way that we go about it is very different. But the problem is that people often conflate these two things, and you read quite a lot of the literature, and you get all these pejorative comments about essentially people spending their time sort of flower pressing and this sort of thing. So we have, I suppose the most famous one of those is in the early 18th century. You have Addison saying that, describing essentially the role of the curator of a herbarium collection as being a sort of learned gentleman who spends his life gathering together the refuse of nature, and it's that disparity between something that is most people see as a pastime to something that's actually a legitimate sort of professional activity, which has, as we've just been talking about, lots and lots of different ways of being used by all sorts of different people.
Oh, well, I'm very glad to hear and learn how important it is, because I think that's fantastic. I think it's great both as a hobby and as a scientific endeavour.
Absolutely.
Well, thank you so much for sparing the time to talk to me. I found it really interesting and inspiring as well, and I want to find out more and read more about it. It's sort of so closely connected to what I do, but completely far away as well. So that's
yes. But fundamentally the techniques are the same.
Yeah.
And as we said right at the beginning, they're essentially all relying on these structural properties of plants to be preserved.
Yeah. Brilliant. Well, thank you so much, Stephen, for sparing the time. And
Pleasure.
thank you very much.
Right. Thank you. Right. Bye-bye.
Bye.